is Vivek Shenoy, who's joining us from the University of Pennsylvania. Very happy to introduce him. And actually, I'd rather get out of the way so we can give more speaking time. So Vivek, please take it away.
Yeah, thanks a lot. So very glad to present at John's 60th birthday. So John and I go way back, probably 15, 20 years. And we published quite a few papers. So here are a few examples of papers on phase field methods, applying them to growth of 2D films. And we worked on a number of other problems as well. And I've shared grants over the past, I would say, 15 years. And I realized that our most recent grant, expired a year ago. So we should fix that. So John, I'll be sure to visit you next year and continue working together. So happy 60th birthday and wishing you many more to come. I should also point out that in addition to science, John has been a great friend. So, when I was moving to Penn from Brown, which is now 13 years ago, I talked quite a bit on a regular basis to John about getting a startup package and about the move, etc. John has a lot of experience. He's been a department chair. So in addition to being a great collaborator, he's been a great friend as well. So today the topic that I've chosen is to understand how forces from the cellular microenvironment make their way to the nucleus, and how that results in changes in the way chromatin is organized or DNA is organized. And so this is, a lot of it is based on phase field method. So it kind of goes back to some of the things we've done. So just to start, if you look at different organs in the body, right, the most important one, perhaps the brain, it's very soft, a consistency of like tofu that you scramble. It's like 1 kilopascal. The stiffest is the bone, more like brick. So you have 6 orders of magnitude in stiffness, when you go from brain to the bone. And now why is this important? So if you look at a triple negative breast cancer cell in its native environment, you have a stiffness of say 500 Pascal. And when it escapes the tumor and goes into the stroma, the stiffness goes up. Then it has to make its way through the circulation, and then it could end up in the bone where it is really stiff, or it could end up somewhere else where the stiffness is not that high. So we are currently part of the Metastasis Research Network, an NCI project, where we're really looking at how, as the cells move from the primary organ to other organs, what the cells have to do is really adapt. And an important part of that adaptation is adaptation to the mechanical environment. And that absolutely involves a change in chromatin organization. So I'll start by explaining how sort of forces are generated in cells when they attach to the extracellular matrix. So this is going to be a reductionist approach, where we are simply going to plate cells on 2D substrates that have the stiffness of the appropriate organ, right? And what we will see is that there's a significant change in the adhesions in the cytoskeleton and also in the nuclear shapes. Now, what's important, what we have discovered is that the cytoskeleton can also lead to epigenetic regulation. That is, it can sort of control which parts of chromatin are open and closed. So I'll talk about the cytoskeleton first, and then how it could potentially regulate epigenetics. And then I will talk about some recent work where we look using high resolution microscopy into the nucleus. And depending on whether the substrate is stiff or soft, there are significant differences in the way chromatin organizes. Here, red is condensed chromatin, which we've been able to model with a phase field approach. And also dynamics. That is, if you stiffen the network, if you stiffen the substrate, then we can actually, in time, see how chromatin organization evolves. So that's the main thing I want to discuss. And more recently, we've been actually going beyond phase field to a polymer approach based on omics data. So while the phase field gives you sort of a mesoscale picture, making sure that we have omics data can actually help us pinpoint which genes are getting up and down regulated. So hopefully I'll have time to go into this. Now, what is the simplest way you can keep everything same, but only change the stiffness that the cells are seeing? So here you have glass, and on that you put a hydrogel of different thickness, and on top of that you can put a mesenchymal cancer cell, say MB231. Now the ligand density is the same on the surface. So in this case, because you have a thick hydrogel which is much softer than glass, this cell will see a soft environment, this will see a stiffer one. And you can see there are significant changes in the nuclear height. And as you can see here, when the cell is on a stiff substrate, it is elongated. And in fact, the adhesions are all at the end, and your actomyosin fibers or cytoskeleton will go over the nucleus and squeeze it down. So the nucleus that you see in the stiff substrate, or you can also change the shape of the cell, either way, is more of a pancake, not the standard round thing that you see in textbooks. That is the case in soft. So now the question is, how does this change the way chromatin is organized? So for this talk, only two concepts are important. The first one being that chromatin is organized in two phases. One is a condensed phase, heterochromatin, that you see as dark regions in this very old electron micrograph, and open regions or euchromatin, where this segment that you see here is accessible to the transcriptional machinery. Now, how do you go between these? So this purple line that you see, that is the double helix, the DNA strand, and it is wound around histones. And when the histones are decorated with acyl groups because of interactions, your chromatin will open up. And when they are methylated, they will close. So these are the only two phases we are going to be considering. And what controls this opening and closing? They are controlled by epigenetic regulators. So for this talk, histone deacetylase is an important one, where you deacetylize and then you can put the methyl marks on it. So simply by these reactions, you can go from one to the other, right? And these epigenetic regulators are controlled by ATPases and other active processes. So now let's look at how in MDA MB231, how the chromatin organization is different when you plate the cell on glass or a stiff substrate or soft substrate. Again, I just want to point out that this is general result. It holds for any type of for almost all the type of mesenchymal cells that we've looked at. So if you look at the stiff substrate, what you see that if you look at the soft substrate, the interior domains are sparse and of larger sizes compared to the stiff substrate. And quite a bit of chromatin is stacked up against the nuclear lamina. They're also called LADs, lamina-associated domains. So the question that we want to ask is, when you look at this picture, it's pretty clear that there are characteristic sizes of the domains in the interior as well as the thickness of the nuclear lamina. So the question is, what gives rise to these length scales? So the first thing to do is, how are the cells adapting in the way of, the cytoskeleton, as I showed you earlier. So here, the basic idea is that this is the cell membrane. That's the inside of the cell. That's the outside. And in response to the forces that are transmitted from the adhesions, couple of things can happen that can sort of lead to this signaling cascade, called Rohrach pathway, where depending on the tension, these the adhesion complex can be stretched, which can then lead to this signaling pathway, which can upregulate contractility. And similarly, increased membrane tension can lead to more calcium, and that can also upregulate contractility. So what we do is we treat the contractile elements as force dipoles, and the main idea is that when there is a mechanical stress, the contractility is it's an isotropic part plus a part that depends on stress. So when the stress is anisotropic, your contractility will also be anisotropic. And this alpha here, in a very coarse-grained manner, describes all of these signaling process. So we've used this to study many different problems: cancer cell motility, macrophage motility, and as well as a number of other problems. So for us, when the cell is elongated, you start off with isotropic contractility. Because the shape is anisotropic, the stress is anisotropic, which will feed back. And what you see here is that the stress fibers or the contractile elements are also anisotropic, and that agrees very nicely with confocal imaging of the cytoskeleton and the adhesions. Now, why is this important? The reason it is important is when there are two sort of things that can happen when G-actin or globular actin sort of polymerizes to become F-actin. There may be molecules that attach to G-actin. So when they polymerize and form F-actin, these molecules can be released and they can go into the nucleus. So MKL, YAP, these are all those examples. But the other thing that can also happen is there could be molecules that can have substrates on F-actin. So they can come out of the nucleus. So histone deacetylase, the molecule that I mentioned, the epigenetic regulator that I mentioned earlier, that falls in this category. So when you form stress fibers, it can come out, okay, and that's going to change chromatin organization. So again, long story short, we can write these reaction diffusion equations, etc. And what we can do is we can change the cell shape. And when you change the cell shape, you change contractility, and when you increase contractility, the histone deacetylase, the nucleus comes down. So the story that we have is that substrate stiffness changes actomyosin contractility, which changes the shuttling of epigenetic regulators. And histone deacetylases are upregulated, and there's also, I don't want to go into too many details. EZH2 is another molecule in this pathway. So both are upregulated on soft. So on soft, you'll have condensed chromatin. On open, you'll have on stiff, you'll have open chromatin. But the question is, what exactly gives rise to these sizes? So here, what we do is we use a phase field approach, where at every point in space, we say that there are two phases of chromatin, euchromatin and heterochromatin, and the remaining is water. So it's a three-phase system, and what we do is we introduce chromatin-chromatin interactions, chromatin lamina interactions. There are three phases, but there is one constraint. So there are two independent degrees of freedom. More importantly, what we do is we account for the interconversion of heterochromatin to euchromatin through acetylation and methylation reactions. Okay, so now if we want to do time evolution, so first you need a free energy. And so we choose two variables. One is the amount of water, which is 1 minus the amount of DNA. So that's one variable. The other variable we introduce is the difference between hetero and euchromatin, kind of like autoparameter. So when this number is close to 1, it's heterochromatin. When it is negative, it's euchromatin. So then what we do is, again, I don't want to go into the details here. This can be obtained using active matter theory or also Flory-Huggins and things like that. So what we do here is we assume a free energy that as a function of these two variables that has two wells, right? One of the wells is at high water content and the autoparameter is negative. So that is this euchromatic phase. The other phase is low water content and the number is close to 1. So that's the heterochromatic phase. So you have three components, two phases, okay? And typically in a cell, you're as far as the water content is roughly 50%, and you may be 0.2, 0.3, you may be somewhere here. So things have this phases have to phase separate, and we are really interested in time evolution. Again, here we are including active force and elasticity. We can put it back, but I don't have time to go into the right questions. I can come back to that. All right, great. Now the question is, how do we evolve the system? So as I said, heterochromatin is a compacted phase. Euchromatin is a swollen phase. So when you convert one phase to the other, there has to be water transport. And this is simply describing the poroelastic effect. The next is heterochromatin likes heterochromatin, euchromatin likes euchromatin. So if you start off with a configuration like this, it would want to go to a configuration like what I've shown here, where you're conserving the net amount of hetero and eus separately. So that gives rise to a term like this, energy-driven term. But the epigenetic reactions do not conserve. They conserve the total amount of DNA, but they do not conserve individually hetero and euchromatin. So you got to write a term here that looks like this, and actually that breaks detail balance. So now what we can do is, if there are no reaction terms, and if you're in a phase like this, we know exactly what will happen, right? You'll get spinodal decomposition, and then you will immediately get coarsening. But now once you put reactions, what you see is that the system sort of evolves and picks a particular size, as well as you can see the formation of these lamina-associated domains. Now, what picks the size? So the reactions will tell you what the homogeneous state is, right? The amount of hetero versus U is decided by the reaction, and also the amount of water, right? So you're in this point in the phase diagram. Now, what you can do is you can stay there, or instead, what you can also do is you can nucleate a phase of heterochromatin here, right? You can do that. And immediately outside, you get this phase, and far field, you're here. So what that will do is it will drive diffusion that will lead to the growth of this. So that will drive diffusion. But this state itself is not stable in the sense that the acetylation reactions will take the red out of this phase. So now the diffusive flux and these reaction fluxes will sort of compete, and they will pick a characteristic length scale. Now it's very easy to write down without doing even any calculations what that length scale is. So you have diffusion divided by the reactions, right? And these factors. Diffusion is like meter square per second. Reaction is 1/second. Take a square root, you get the size. So basically the size here is simply decided by what the methylation rate is. And we know that on stiff substrate the methylation is small, so you will get small domains. And on soft substrate it's high, so you'll get bigger domains. And so this is only an approximate theory, a dilute theory, but when you do the phase field calculations, it actually works out pretty well. You can come up with corrections to this, which we've done, but I won't go into this. So the scaling works out very nicely. Now, so here, what I didn't do is I didn't consider the nuclear lamina. So now when I go and now what we say is that every segment of DNA can interact with the nuclear lamina. Now, so this is similar to a case where water molecules can have attractive interactions with glass. But on a cold day, you will get water condensing, or you'll get, maybe little ice crystals on the surface. The reason is the denser phase is the one that will condense. Reason being that it's got higher density right? So this is exactly the same thing. So we assume that there are attractive interactions between chromatin and the nuclear lamina. And when you do that, and if you keep the epigenetic reactions the same and keep increasing chromatin lamina affinity, you get this wetting transition where you go from sort of a finite contact angle to to zero contact angle where it fully wets. But that's not the only thing that controls the thickness. Just as the size of the interior domains is controlled by the methylation reaction, same physics also controls when you increase methylation. You can get higher chromatin lamina affinity. So both the chromatin lamina affinity and methylation will control the shape and size of these lamina-associated domains. And in fact, when we go back to our experiments and look at it, when there is more thickness of the chromatin lamina-associated domains, you'll get higher sizes of interior domains as well. So that agrees very well. Now, how do we test if this is correct? Now, instead of stiffening the substrate, if we keep the substrate, if we keep the cell on a soft substrate and treat it with an epigenetic regulator GSK, which actually prevents methylation. So it is exactly like going to a stiff substrate, and you can see in this case, it does not completely recapitulate the stiff configuration, but you're getting closer there. So, and the other test we can do is we said that it is it is these contractile elements that are getting HDAC out. So if you inhibit contractility, you get similar behavior, that you get these chromatin lamina associated domains. Now, the other interesting thing that we found is from the theory, when we extract the strength of the chromatin lamina interactions, we get two peaks actually, one at high affinity and the other at low affinity. The reason is that in the nucleus, there can be nuclear pores. So nuclear pores have very low affinity. So we are able to actually recapitulate a lot of interesting biology using this simple phase field approach. Okay. So the next thing I want to talk about is dynamics. So here, what we can do is, so let's say a cell, leaves an organ, and in a few days, it's going to go find its home somewhere else. Now, to simply, in a reductionist approach, if you want to capture what's happening, what we can do is we can put the cell on a soft substrate. And then with light, if you shine light, that's going to cross bonding. That's magic. That's chemistry that Jason Birdie can do. So this cross-linking happens actually quite fast. And then we can actually look at chromatin and see how it remodels in time. So you see that on a stiff substrate, you have more lamina-associated domains, these bigger domains here. And in time, you see that these radii decrease, your lamina-associated domains go down. So the heterochromatic diameter decreases, the domain number increases, and the spacing decreases as well. So now, since we have dynamics, we can understand how this happens. So here is a movie where we start off with why is this not running? Okay. So right. So if we simply evolve it, we are not going to get what the experiments say. You are also nucleate. So we randomly nucleate domains. And as you nucleate domains, you see that there is reorganization, and then you'll reach a point where you cannot nucleate further. So if you look at the reaction rates are kind of in the 1 to 10 minute time scale. And the diffusion between domains is also in the same range. But nucleation can be a really, really slow process. So we believe that it's the nucleation of these new domains which give you sort of these larger number and smaller scale, smaller spacing that is governing the dynamics. And as you know, nucleation is the hardest thing to understand in soft matter physics or in solid state physics. It's the same here, right? Because many things have to conspire for the nuclei to form. So we are now carefully studying nucleation. This I will skip. So in the last 2 or 3 minutes that I have, I just want to talk about how we can go from this mesoscale approach to really predicting which genes are upregulated or downregulated. So here, what we do is, we use this ChIP-seq data, which tells you which proteins can be found on every segment of DNA. And with 10 kilobase pair resolution, what we do is we use sort of a principal component analysis to assign every segment of DNA to euchromatin or heterochromatin. Then, just as in our phase field model, we include diffusion of water, diffusion of epigenetic marks, and reactions, which no polymer model to our knowledge does. So we include that. And now we can see that absolutely, as a theory predicts, when you change acetylation or when you go from stiff to soft, you're going to change this domain size, nicely agreeing with our predicted theory. Now, what's nice about what's attractive about this is that you can treat this with epigenetic drugs, which can say open up a chromatin. And again, we can compare the data with experiment. Now, the main point here is that there is also this technique of high C, which actually tells you in 3 dimensions which segment of DNA is sort of touching the other segment. So this is sort of this contact map that you see here. And so when you treat it with this drug that opens up DNA, right, the main point is that all the changes are occurring at the interface of this hetero and euchromatin. So, right, so all the compartment changes, that is, this hetero to you, all of them are happening at the boundary. And in fact, surprisingly, if you look at all the important genes that are necessary for EMT, say the MAP kinase or Wnt, or ERK, all of them are located very close to the boundary. So there's a very important principle of this organization where, where the housekeeping genes may be elsewhere, but the relevant genes are actually placed next to these domain boundaries. So when you change stiffness or treat with a drug, all that's happening is, you're just opening and closing what's happening near the boundary. So to summarize, what I showed you is that chromatin organization depends on stiffness and the cytoskeleton contractility. The heterochromatic domains regulate gene expression as revealed by combining omics data with polymer simulations. And also, once you have this nucleation, that means you're embedding a memory. So when the cell leaves an organ, goes somewhere else. There can be permanent memory that can be imported to chromatin, and that's something we are studying in the context of metastasis. So I want to finish by thanking the folks who did the work. So a lot of the face field work was done by Ayushkant, and as of last 3 years, we have a wet lab, and a lot of the imaging that I showed you is from our wet lab. So Zhijian Guo, he's involved in imaging, and Vinayak is involved both in imaging and polymer simulations. With that, I'll be happy to take any questions.
Thank you very much for a wonderful talk. We have a couple minutes for Q&A before we turn over to Miriam for her